Caleb has had a really incredible treatment journey. He was diagnosed as a young child with a low-grade glioma. Low-grade gliomas are the most common type of brain tumor that we see in children. But then years later, when our team met him, he now has all these meningiomas. So we think they're radiation-related sort of secondary tumors.
Kids are generally very resilient. They tolerate a lot of therapy, but they're also at a very vulnerable point where their brains are developing. So you can't really radiate them at really young ages without having some long-term effects. Over the past decade, we've made huge advances in understanding how brain tumors function, what makes them tick. So now our job is, we understand the biology, but how can we treat these children better?
Also a consequence of what we found that he has a genetic predisposition to tumors. It's part of what our Institute for Genomic Medicine is able to offer. And now, really excitingly, we're able to offer it to all of our brain tumor patients. Now at diagnosis, all to further characterize their tumor, but also learn about any genetic risks for tumor development.
So this has been a huge effort where the biology, the genomics, and those aspects of this are going to be critical for us to then come up with better therapies for these kids.
Even from the beginning, they were excited to be a part of sort of something new and evolving. There's a lot of things that they've done and are sort of for a research purpose, but they believed in it. Connect 2007 is an early phase clinical trial. Specifically, it's a type of targeted radionuclide therapy, so you can almost think of it as a type of targeted IV radiation. So the thought for someone like Caleb is that this treatment, this targeted radiation, should target the tumor cells that have the somatostatin receptor, but spare the healthy cells. This was brand new to everyone, and so there was something we learned each time. We were able to incorporate that going forward.
We need some of these targeted therapies that will come from understanding the biology and the genomics, because that allows us to then personalize therapy for that patient. And I think this is why this next decade is such an exciting decade for us, because that's exactly what we're working on.
The bell ringing was a celebration of finishing all those 6 cycles, getting through the trial treatment, having stable imaging in terms of his meningiomas and shrinkage. And I think also a celebration that he was the first patient, that we learned so much from him. So this was sort of a celebration of all that he's taught us too.